Today we are wading into something completely different. We are looking at what I call the accidental killers, the ones that, frankly, don't really care if we exist or not. They are in the soil outside your window, they are in the river you swim in, they might even be on the flowers in your garden right now.
And that indifference is, paradoxically, exactly what makes them so dangerous. We are talking about the opportunists.
Specifically, we are doing a massive deep dive into Chapter 16 of our microbiology source material. We are focusing on a group of bacteria that I like to call the hospital nightmares. We are talking about Pseudomonas and its very dangerous friends.
It is quite the rogues gallery. We are dealing with gram-negative rods, but friends is a loose term here. It is more like a criminal syndicate where everyone has a specific specialty. You have the leader, Pseudomonas aeruginosa. You have the potential bioterrorist candidates, the Burkholderia species. You have the scavenger, Stenotrophomonas, and finally the soldier, Acinteobacter.
And the source material highlights a really disturbing paradox right off the bat that I want to get into. These bugs are everywhere. I mean, literally everywhere in nature. But they rarely hurt healthy people.
Correct. If your immune system is intact, you are generally safe.
But if you step into a hospital, the one place in the world designed to make you better, these are the things that are most likely to kill you. So what is our mission for this deep dive?
Our mission is to understand the mechanism of opportunity. We need to understand how a bacteria that is perfectly happy eating rotting organic matter in a mud puddle evolves the machinery to dissolve human lungs. We are going to look at their weapons, their armor, and why, despite our best antibiotics, they are currently winning the war in our ICUs.
It is the kick you when you are down philosophy of microbiology. Let's get into it.
Let's do it.
Before we meet the specific characters, we need to understand what binds this group together biologically. The text calls them non-fermenters. Now, for the non-microbiologists listening, that sounds like a brewing term. Why does that matter in a medical context?
It matters because it defines their lifestyle and how we identify them. Think about the bacteria in your gut. The Enterobacteriaceae, like E. coli or Salmonella, they live in the intestines, which is a dark, oxygen-poor environment. To get energy, they ferment carbohydrates. They are built for fermentation.
Okay, so they turn sugar into energy without needing a lot of fresh air.
Oversimplified, but yes. The group we are discussing today, Pseudomonas and its cohorts, are different. They are generally strict aerobes. They do not ferment. They respire. They use oxygen to generate energy via an electron transport chain, just like our own cells do.
Which means they cannot really live deep in the gut.
Generally, no. They aren't gut bugs. They are environmental bugs. They are what we call saprophytes.
Saprophyte.
It is a fancy way of saying they eat dead stuff. Decaying vegetation, organic matter in soil, sludge in water. They are the recyclers of nature.
So they are the composters.
Exactly. And because they are environmental, they are incredibly tough. Think about life in a puddle or in the topsoil. The temperature changes constantly. It freezes, it burns. There are other bacteria trying to kill you. There are natural toxins. If you can survive in the soil, you have evolved a level of durability that fragile gut bacteria just do not have.
And that durability translates to the hospital environment.
It translates perfectly. Because they are aerobic and environmental, they love moisture. Sinks, showers, humidifiers, respiratory equipment, mops, buckets, that is their playground.
There was a detail in the reading that really surprised me. We always hear about the human microbiome and how we are covered in bacteria. I assumed Pseudomonas was just part of the mix hanging out on my arm right now.
That is a very common misconception. Pseudomonas aeruginosa, the dangerous one we are focusing on, is almost completely absent from the skin and nose of a healthy person.
Really? Even though it's in the water I shower in?
Yes. You might have other harmless Pseudomonas species on you, but aeruginosa, it is a transient visitor at best. It tries to land on you, and your skin, specifically the fatty acids secreted by your skin, tells it to get lost. It simply cannot get a foothold on healthy, dry skin.
So if I am healthy, I have a biological no trespassing sign up that specifically targets this bug.
Precisely. But if you strip away that defense, if you have a burn that destroys the skin or a wound or a plastic tube inserted into your vein or bladder, that sign comes down. And that is when the environmental survivor decides to move in.
It sees the open door. Okay, let's talk about the ringleader, the big bad wolf of nosocomial or hospital-acquired infections, Pseudomonas aeruginosa.
The leader of the pack. Under the microscope, it's just a simple, motile, rod-shaped bacteria. Very small, maybe 0.6 by 2 micrometers. It occurs singly, sometimes in pairs or short chains.
The name itself breaks down into something quite descriptive, doesn't it?
It does. Pseudomonas roughly translates to false unit, referring to its shape. But aeruginosa is the interesting part. It refers to verdigris, or the rust of copper, that specific blue-green color you see on old statues.
Now, that color isn't just a poetic name. It is a major identifier in the lab.
It is the calling card. It forms these smooth, round colonies. But before we get to the color, this brings us to the sensory experience of this bacteria. Usually microbiology is very visual. We look at shapes under a microscope. But Pseudomonas, you can often identify it with your nose.
This was my favorite part of the chapter, the smell test.
If you walk into a microbiology lab and someone has opened an incubator full of Pseudomonas cultures, you know it instantly. It has a heavy, sweet scent.
The text says grape-like.
Grape-like is the classic description. Some people say it smells like cheap artificial grape soda. Others, interestingly, say it smells like corn tortillas or tacos.
Corn tacos, that is wildly specific.
It comes from a specific chemical compound they release called aminoacetophenone. It is a very distinct olfactory signature, but sadly it has a dark side. If a patient has a severe burn infection with Pseudomonas, you can sometimes smell that sweet grape scent coming from the wound itself.
That is a haunting image, the smell of infection being sweet.
It is for clinicians. It is a smell you don't forget.
Let's go back to the visuals. You mentioned the verdigris, or copper rust color. This bug essentially paints its environment.
It is an artist.
Yeah.
Most bacteria, when you grow them on a petri dish, are just beige or off-white slime. Pseudomonas produces vivid pigments. There are two main ones you need to know to understand this bug.
Walk us through them.
First, you have piocyanin. Cyan implies blue. This is a blue pigment that is water-soluble. It seeps out of the bacteria and stains the agar jelly in the petri dish, or the bandages of a patient, a bright blue.
And the second one?
Pioverdine. Verde implies green. This one is fluorescent. If you shine a UV light on it, it glows a bright yellow-green.
So you mix the blue piocyanin and the yellow-green pioverdine.
And you get that characteristic bright blue-green color. It is unmistakable. I should note, the text does mention some rare variants. Occasionally, you'll see strains that produce a dark red pigment called pyorubin or a brown-black one called pyomelanin. But the fluorescent group, the blue-green ones, those are the primary troublemakers.
Is there a purpose to the color? Or is it just evolutionary decoration?
Oh, in biology, nothing is just for show. Take pyoverdine, the green one. It acts as a siderophore.
A siderophore. Let's unpack that term.
It literally means iron carrier. Iron is essential for life. Bacteria need it to breathe and multiply. But in the human body, we know this, so we lock all our iron away inside our cells or bind it tightly to proteins like transferrin. We don't leave free iron floating around precisely so bacteria cannot eat it.
We starve them out.
Right. So Pseudomonas secretes pyoverdine like a little retrieval drone. It goes out into the environment, rips iron molecules away from whatever is holding them, and brings them back to the bacteria. It is a theft mechanism.
So the green glow is actually a survival tool. What about the blue one, piocyanin?
That is a weapon. Piocyanin generates what we call reactive oxygen species, or free radicals. It causes oxidative stress and stimulates inflammation by triggering the release of interleukin-8, which attracts immune cells to the area.
We hear about free radicals in skincare commercials as being bad for aging.
Exactly. They damage cells. Pseudomonas uses this blue pigment to generate toxic chemicals that damage host cells and even kill off other competing bacteria in the wound. It is toxic chemical warfare, and it happens to be blue.
Incredible. And just to round out the lab profile, the source mentions some key biochemical traits, like growing at high temperatures.
Yes, it grows very well at 37 to 42 degrees Celsius. That ability to grow at 42 degrees is a key test to distinguish Pseudomonas aeruginosa from other less harmful fluorescent pseudomonads in the environment. And it's oxidase positive, which is a big differentiator from the gut bacteria and from Stenotrophomonas, which we'll get to later.
So we have a grape-smelling, blue-green, iron-stealing bacteria that likes it hot. But it doesn't just sit there. The source material outlines a terrifying arsenal of weapons. It seems like Pseudomonas has a tool for every occasion.
It does. It has a massive genome compared to other bacteria, and a lot of that code is dedicated to weaponry and adaptability. But before it can use those weapons, it has to stick.
The attachment phase.
Right. Imagine a fast-flowing river. If you want to live in the river, you need to grab onto a rock. In the body, that river is our flowing blood, or the movement of mucus and cilia in our lungs. Pseudomonas uses appendages called pili, or fimbriae. They are like microscopic grappling hooks that attach directly to host cells.
So it hooks onto a cell, then what?
Then it builds a bunker. This is one of the most important concepts in modern microbiology, the biofilm.
We hear this term biofilm a lot. It sounds like something you scrub off your shower tiles.
That is a biofilm. The slime on your shower tile is likely Pseudomonas or relative, but in the body it is devastating. The bacteria secrete a slime layer, an exopolysaccharide called alginate. Think of alginate as concrete. The bacteria secrete this goo and they live inside it. It connects them together into a community.
A microscopic city.
Exactly. And this city has walls. White blood cells cannot chew through the alginate effectively. Antibodies get stuck in the slime. And most importantly, antibiotics cannot penetrate it well.
So the bacteria are sitting inside this bunker, safe from our medicines, and they are launching attacks.
Correct. And the coordination of that attack is fascinating. They use something called quorum sensing.
Quorum sensing, like a voting quorum?
Yes. Bacteria can count. They secrete little chemical signals. When the bacteria are few in number, the signal concentration is low, they stay quiet, they hide.
Sneaky.
But as they multiply, the level of that chemical signal rises. When it hits a specific threshold, they know, okay, there are enough of us here now, and instantly they all turn on their virulence genes at the same time.
So they wait until they have an army and then they strike all at once.
It is a coordinated ambush. If one bacteria released toxins, the immune system would spot it and squash it. But if a billion release toxins simultaneously, they overwhelm the defenses.
That is frighteningly intelligent for a single-celled organism. Let's talk about those toxins. The text mentions endotoxins like lipopolysaccharide or LPS.
LPS is standard for gram-negative bugs. It's part of their outer shell. When the bacteria die and break apart, the LPS is released and causes massive immune reactions. Fever, shock, sepsis.
But then there's the more targeted stuff, exotoxin A.
Exotoxin A is a heavy hitter. It causes tissue necrosis. It functions almost exactly like the toxin causing diphtheria, which is one of the most potent toxins known.
How does it work on a cellular level?
It enters your cell and locates the ribosome, the factory that builds proteins. Specifically, it chemically modifies a factor called elongation factor 2 via a process called ADP ribosylation.
That sounds technical. What is the analogy?
Imagine a zipper. Elongation factor 2 is the slider that moves the zipper up. Exotoxin A jams the slider. The protein chain cannot be built. The factory stops.
And if a cell cannot make proteins?
It dies.
Right.
Rapidly. And then there are the injectable toxins. The text mentions exoenzymes S and T.
This involves the type 3 secretion system, right?
Yes, and this is incredible engineering. The bacteria builds a molecular needle, a microscopic syringe on its surface.
A literal syringe.
A literal syringe. It punches a hole directly into the membrane of your cell and injects poisons like exoenzyme S and T directly into the cytoplasm.
It injects them inside our cell.
Yes. These enzymes scramble the cell's internal skeleton, or they interfere with the immune response to stop the cell from eating the bacteria. It effectively paralyzes the immune cell from the inside out.
And finally, elastase.
Elastase does exactly what it sounds like. It breaks down elastin. Elastin is the protein that makes your tissues stretchy and holds them together. It is in your blood vessels, your lungs, your skin.
So by breaking that down,
it literally dissolves the glue holding your tissue together. This allows the bacteria to chew through blood vessel walls and spread deeper into the body, causing hemorrhage and destruction.
we have got the weapons. Now let's see the damage. What does a Pseudomonas infection actually look like clinically?
Well, for the healthy listener, there are two superficial infections you might actually encounter. They are annoying, but usually self-limiting. We call these the recreational infections.
Let me guess. Hot tub folliculitis.
Exactly. This is the classic Monday morning rash. You go away for a weekend, stay in a hotel, jump in the hot tub.
And the maintenance guy didn't check the chlorine levels.
Right. Hot water breaks down chlorine faster than cold water. And the swirling water, Pseudomonas loves it. The turbulence forces the bacteria into your hair follicles. A day or two later, you have this itchy, bumpy red rash everywhere the water touched.
Gross, but not deadly.
Rarely. Usually it goes away on its own if you stop using the tub. The other one is swimmer's ear, or otitis externa. Water gets trapped in the ear canal, the bacteria grow in the soggy skin, it gets inflamed. Painful, but treatable.
But we aren't here for earaches. We are here for the deep dive stuff. The deep infections, meningitis from neurosurgery, urinary tract infections from catheters, necrotizing pneumonia from contaminated respirators. But the text describes a specific skin condition called eczema gangrenosum.
This is the nightmare scenario. This happens in immunocompromised patients who have Pseudomonas in their blood. Severe sepsis.
What does it look like?
It is a skin lesion, but it is not like a pimple or an abscess. It is a patch of hemorrhagic, dead tissue, necrotic skin surrounded by a bright red, angry ring.
And the text makes a specific point. No pus.
That is the key diagnostic clue. Normally, infections have pus, that's white blood cells fighting the battle. In eczema gangrenosum, there is no pus because the bacteria has invaded the walls of the blood vessels supplying that patch of skin and destroyed them with elastase.
It cut the supply line.
It infarcted the skin. The tissue died because it was starved of blood. It is a sign that the bacteria is running rampant in the bloodstream and destroying vasculature.
That is incredibly stark. But there is one group of patients where Pseudomonas is truly the defining enemy. Patients with cystic fibrosis.
This is a tragic and long-standing battle. Cystic fibrosis, or CF, is a genetic condition where the lungs produce thick, sticky mucus.
And Pseudomonas loves mucus.
It views it as a perfect habitat. Once Pseudomonas gets into the lungs of a CF patient, usually in childhood or adolescence, it is almost impossible to eradicate. It is a major cause of morbidity and mortality for them.
Why? Is it the biofilm again?
It is the biofilm on steroids. The text mentions that strains isolated from CF patients are often mucoid. They undergo a genetic switch over time.
That's a witch?
Yes. When Pseudomonas first arrives, it might be the normal wild type. But under the pressure of the lung immune system, it mutates. It turns on the alginate genes full blast. It becomes incredibly slimy.
So they coat the lungs in slime.
They create a thick, impenetrable layer. It blocks the airways, it causes chronic inflammation, chronic pneumonia, and it slowly destroys the lung tissue over years. It is a chronic, lifelong colonization.
We have to talk about treatment. Because everything you've described, the biofilm, the toxins, makes me think a simple penicillin is not going to cut it.
A simple penicillin would just make Pseudomonas laugh, if it could. This bacteria is largely defined by its resistance. It's intrinsically resistant to many drugs.
Is it resistant because we have used too many antibiotics?
Partially. But Pseudomonas is intrinsically resistant. Remember, it lives in the soil. The soil is a chemical war zone. Fungi are producing molds, which are basically penicillin factories. Other bacteria are producing streptomycin. Pseudomonas evolved defenses against antibiotics millions of years before humans discovered them.
So it was born wearing armor.
Exactly. It has three main layers of defense. First, altered cell wall channels. It has porins, little channels that let food in. Pseudomonas can close these channels or change their shape so the antibiotic molecule is physically too fat to fit inside.
Denied entry.
Second, it has enzymes like beta-lactamases. If a penicillin molecule manages to get inside, these enzymes chop it into pieces before it can do any harm.
Active destruction.
And third, and most impressively, it has multi-drug efflux pumps.
I love the name, efflux pumps.
Think of a bouncer at a club. The antibiotic sneaks in, the bouncer spots it, grabs it by the collar, and physically throws it back out the door. Pseudomonas has pumps that run constantly, bailing out drugs.
So how do doctors fight it? Especially since it's the 5th most common pathogen in hospital-acquired infections, making up like 10% of cases.
We have to be strategic. The golden rule for treating serious Pseudomonas infections is combination therapy. You usually need 2 drugs to prevent resistance from developing right in the middle of treatment.
Because it will adapt.
Yes. If you use one drug, the bacteria might mutate its porins and become resistant during the treatment. So we hit it with 2 drugs that work in different ways.
Give us an example combo.
We might use an extended-spectrum penicillin, like piperacillin, which disrupts the cell wall, and we combine it with an aminoglycoside, like tobramycin, which attacks the ribosome.
A 1-2 punch.
Right. Or we use the specific drug options like aztreonam, fluoroquinolones like ciprofloxacin, cephalosporins like ceftazidime or cefepime, or the heavy artillery, the carbapenems like imipenem or meropenem. But even these are failing. Prevention is almost more important now. Keeping hospital equipment, sinks, showers, completely dry.
It sounds like a constant arms race. Okay, that is Pseudomonas, a formidable enemy. But we promised a deep dive into the friends. And honestly, reading the text, the friends are just as scary, if not more so in their specific niches. Let's move to the Burkholderia species.
Burkholderia. These used to be classified as Pseudomonas, but genetically they're distinct enough to get their own genus. They are the dangerous cousins. Small, gram-negative rods.
I want to start with the one that sounds like a spy novel plot, Burkholderia pseudomele.
Ah, the agent of melioidosis, also known as Whitmore's disease. This is a fascinating and terrifying organism.
Where do we find it?
It is tropical. We find it mainly in Southeast Asia and northern Australia. It lives in the soil, fresh water, and specifically rice paddies. It's a true saprophyte.
And the infections peak during the monsoon season, right?
Yes. Heavy rains churn up the soil. People get infected from inhaling dust or getting contaminated soil and water into cuts. And under the microscope, it has this distinct bipolar staining. It looks almost like a safety pin. And the colonies become rough and wrinkled on the agar.
The text mentions a time bomb aspect to this one.
Yes. It can have a latent period of months to years to even decades. It hides inside your body.
Like tuberculosis.
Very similar strategy. You might get exposed to it, recover from a mild illness, and the bacteria just goes dormant. Decades later, your immune system weakens, maybe from age or another illness, and suddenly it reactivates. Severe primary pneumonia, overwhelming septicemia, abscesses in multiple organs, high mortality if untreated.
That is unnerving. You could just be carrying it. And because of its potential to be spread via aerosol and its high mortality, the government has an eye on this one.
Yes, it is classified as a Category B agent of bioterrorism.
Speaking of bioweapons, let's talk about its brother, Burkholderia malae.
Burkholderia malae, the cause of glanders. Now, pay attention to the name. Pseudomalae means false glanders. Malae is the real deal.
What is the difference between them?
Pseudomalae, the rice paddy one, lives in the environment. Malae does not. It is a parasite. It lives primarily in livestock, horses, donkeys, mules.
And this was actually used as a weapon.
It was one of the first biological weapons in modern history. We go back to World War I.
World War I.
Yes. It was used to infect horses and mules. Remember, armies ran on horses back then. They pulled the artillery, they carried the supplies. If you could get the enemy's horses glanders, you crippled their logistics.
That is diabolical.
It is. It is a horrific disease for the animals. And it can jump to humans who handle the animals through skin abrasions or inhalation. Thankfully, it is very rare in humans now. The last U.S. case was in 1934, not counting the occasional modern lab accident. But it's still a Category B agent.
And there is a key physical difference between these two brothers that helps a lab tell them apart.
Yes. Pseudomalae, the environmental one, is motile. It can swim. Malae, the horse one, is non-motile. That is a big clue for the microbiologist.
Okay, there's one more Burkholderia we need to cover. The Burkholderia cepacia complex.
The complex part is key. It is not just one bug. It is a group of 17 different species, or genomospecies, environmental organisms that can grow in water, soil, and even decaying vegetable materials.
Which is where it gets its name, right? It's known as a plant pathogen that rots onions.
Exactly. Cepacia comes from the Latin for onion. It does a great job decomposing organic material, but in the hospital, specifically for cystic fibrosis patients again, it is a terror. We talked about how bad Pseudomonas is for CF. Burkholderia cepacia can be worse.
Why? Is it the biofilm again?
It is the transmissibility. Pseudomonas usually comes from the environment. Cepacia is highly transmissible from one CF patient to another.
That changes the social dynamic entirely.
It really did. It forces social distancing among CF patients to prevent the spread. They can't be in the same room. It is associated with rapidly progressive deterioration and necrotizing pneumonia. And treatment is incredibly difficult because it's extremely resistant to standard drugs like the aminoglycosides we use for Pseudomonas. We usually have to treat it with trimethoprim, sulfamethoxazole, or TMP-SMX.
It sounds like resistance is the recurring theme of this deep dive.
It absolutely is. Which brings us to our next character.
The specialist. Stenotrophomonas multifila.
That is a mouthful.
Let's break it down. Stenotrophomonas.
It breaks down to narrow feeder, and multifila means malt-loving, free-living, gram-negative rod.
And how do we identify this one in the lab?
Unlike Pseudomonas, it is oxidase negative. It is also positive for lysine decarboxylase, which helps differentiate it. It forms these distinctive lavender green colonies on blood agar, and it has an ammonia-like odor.
Not exactly the sweet grape smell of Pseudomonas. Why do you call it the specialist opportunist?
Because it thrives specifically on our failure. Stenotrophomonas often appears in patients who are already receiving broad-spectrum antibiotics, especially patients with indwelling intravenous catheters.
Wait, explain that mechanism. You are taking drugs to kill bacteria, and this one shows up.
Imagine a garden. You want to get rid of the weeds. So you spray a powerful herbicide, a broad-spectrum antibiotic like carbapenem. All the regular weeds die.
Great, clean garden.
But Stenotrophomonas is intrinsically resistant to that herbicide. It was sitting there, suppressed by the other bacteria. Now that the competition is dead, it has the whole area to itself. It explodes.
So we create the opening for it.
We select for it. The very drug we use to save people from serious infections, like imipenem, is the red carpet for Stenotrophomonas.
And there's a weird resistance quirk here, too, involving heavy metals.
Yes. It is resistant to heavy metals, specifically silver.
Why does that matter in a hospital?
We use silver in medicine. We use silver-coated catheters and silver-impregnated dressings because silver is toxic to most bacteria. But Stenotrophomonas just shrugs it off.
So how do you kill it? If it resists the big hospital drugs like carbapenems.
This is the treatment paradox. While it's resistant to powerful hospital drugs, it is usually susceptible to an older, common drug. Trimethoprim sulfamethoxazole, TMP-SMX.
The same one used for the onion bug, Burkholderia cepacia.
Exactly. The superbug that eats carbapenems is weak against this older drug. It is a strange vulnerability, but it saves lives.
Let's meet our final character, the survivor.
Acinetobacter.
Specifically, Acinetobacter baumannii, ubiquitous in soil and water.
Now, physically, this one looks different from the others under the microscope.
Yes, Pseudomonas and Burkholderia are slender rods. Acinetobacter is what we call coccobacillar.
Which means?
It is short and plump, almost round. Under a microscope, it looks so round it can be easily mistaken for Neisseria, the bacteria that causes meningitis or gonorrhea.
But the key difference is the oxidase test, right?
Right. Neisseria is oxidase positive. Acinetobacter is oxidase negative. That is the crucial lab distinction.
The outline calls this the soldier's infection. Why? This is a direct reference to the Iraq War. We saw serious wound infections among servicemen returning from Iraq, traumatic injuries involving soil contamination.
Okay, so initial contamination from the desert sand.
But then the bacteria gets into the hospital environment, and Acinetobacter has a clinical relevance as a major hospital pathogen, causing ICU pneumonia and catheter infections because it has a superpower that Pseudomonas does not have.
What is it?
Pseudomonas needs water. If a surface dries out, Pseudomonas dies pretty quickly. Acinetobacter can survive on environmental surfaces, dry surfaces, for weeks.
Weeks?
Yes. Bed rails, curtains, medical equipment, it just sits there completely dried out waiting. It makes it a nightmare for hospital infection control.
And the resistance.
It is a multi-drug resistant nightmare, MDR strains. Treatment often requires testing a whole battery of drugs. Carbapenem resistance here is a major public health challenge.
So what do you do?
Sometimes, colistin is the absolute only active agent left for these superbugs. And colistin is highly toxic to the kidneys. We are forced to use a toxic drug because it's the only weapon we have left.
We have met the cast. We have seen the damage. But I want to connect the dots here for a second. We call these nosocomial pathogens hospital-acquired.
Yes. They are environmental survivors that have adapted to the hospital environment.
And listening to you, the defining challenge for all of them, Pseudomonas, Burkholderia, Stenotrophomonas, Acinetobacter, is antibiotic resistance. Efflux pumps, beta-lactamases, biofilms.
Exactly.
It sounds less like bad luck and more like we are forcing them to evolve.
We are. That is the paradox of modern medicine. By creating environments like ICUs and using broad-spectrum antibiotics, we have essentially cleared the field of weak bacteria. We've created a paradise for these tough, resistant environmental organisms to thrive.
We built a luxury resort for the toughest bugs on Earth.
We did. We created a unique ecosystem.
So here is a final provocative thought for you to chew on, building on everything we've talked about today. If these bugs are in the soil and water around us every day without hurting us, what does their rise in hospitals tell us about the fragility of human defenses when high-tech interventions break down our natural barriers? And further, we talked about how these bacteria communicate with quorum sensing. They count each other. They coordinate attacks.
It is a primitive form of intelligence.
It makes you wonder. We treat bacteria like mindless germs. But they have social behaviors. They have strategy. As we push them with more and more antibiotics, forcing them to evolve faster and communicate better to survive, are we inadvertently training them? Are we making them smarter?
That is a very uncomfortable question.
Something to think about the next time you use a heavy-duty antibacterial wipe. Thanks for joining us on this deep dive. Stay curious, and we will see you next time.